Firstly, thank you for talking to us. Just to give our viewers a bit of background, that you have 25 years experience as a researcher and more importantly as an educator. And we are having a crisis of communication along with this health crisis in the world today. And the information about the pandemic is more about, blaming and wondering where the viruses come from, etc. Hence, we find the need to talk to a researcher to understand what the solutions are, because World Health Organization keeps saying, and governments all over the world are talking about vaccines. So you do research in developmental biology, but you also teach about vaccines and immunology. So tell us from your experience and experience of the scientific community, what are the stages in vaccine development, and where on this timeline are we right now with the COVID-19 vaccine?
Thank you, Annabelle, for inviting me for this. So let me start by what we mean by a vaccine and what a vaccine really does. Basically, every time we get infected with any pathogen, usually what happens is our body has a system which we call the immune system, which recognizes that particular pathogen and tries to mount an immune response. Usually what also happens is that once you've been exposed to a particular pathogen, the next time you see the pathogen, you are better able and better equipped to fight the same organism. This is how our body normally fights. And the same idea is used when we talk about vaccine. So the vaccine is an artificial exposure of our body to a particular pathogen. Now, the problem with COVID-19 is that it is a totally different virus, which our world hasn't been exposed to in earlier time. And therefore, nobody has an immunity to it. And hence, it is spreading all over the world. Also, the other problem is the way it spreads is extremely rapid, and that is why we are in this situation. Because there have been other coronaviruses. There was SARS, there was MERS, there were other viruses like Ebola, which did not create this huge pandemic because it had certain characteristics which allowed us to have an upper hand and fight against it. So, if we look at vaccine, basically that means what are we trying to do? We are going to artificially try and use some component of the virus, which obviously cannot cause the disease itself, and inject it into humans, and then let our immune system recognize this and build a response to it. Hence, if you actually get exposed to the virus in the natural environment, your body would be prepared to fight it. Now, the way vaccines are developed, as you can imagine, and you must have read, is a very, very long and complicated process. On an average, to develop a normal vaccine in the normal timeline would be at least 5 to 10 years, which, of course, in our situation, we don't have that time. And of course, because of this fact, the whole world has been affected, and I believe that there are at least 100 vaccines being developed all over the world. We can expect that time is going to be highly reduced. But even when we say reduced, it's probably not going to be anything less than a year to year and a half. So there are 2 or 3 strategies that can be used. One of the things, and that has been the traditional idea, is you use the entire organism, okay? But obviously you can't use the organism as it causes a disease. So either you kill the organism so it won't cause a disease, or you alter it. And alter it sufficiently so that it will still look like the organism that causes a disease, but will actually not be able to cause a disease. We call this attenuation of the organism. And as I said, this was a traditional method that was used. And there are a lot of labs which are trying this method. Viruses, of course, are very difficult to grow because they need a host. So getting enough viruses to actually kill and make a vaccine is very cheap. And it would take a long, long time, which is why people are using other strategies. What are the possible other strategies? Is that you use parts of the virus and let the body recognize those parts, and hence they will be able to recognize the entire organism when they get exposed to the entire virus. So what are the various parts? The obvious thing is that the virus has several proteins on its surface, and everybody must have heard about this spike protein, the one that sticks out from the surface of the virus, which actually is the one that recognizes a cell by attaching to its receptor, and therefore obviously that's the first part. So people are trying to get this protein, purify it, and then use that as a source of vaccine. That itself is not very easy, because again you would have to artificially either make the protein, or you have enough virus particles so that you would extract it. People are using a whole lot of other strategies, and two interesting strategies that are being used is that generally a protein is made by a cell, and that could be any cell including our own cells, is because there is a DNA or a code for it in our genetic makeup. So instead of actually using the protein, a lot of scientists now are using the DNA itself. But the logic is that when a virus enters, so you must remember, a virus is a very small particle. It's a very, very small organism which actually cannot survive on its own because it cannot code for its own protein. So it infects the cell and in fact uses our cell's machinery to make the protein. It has a DNA though. So the logic that is being used for this particular vaccine is that the DNA itself is going to be injected into our cell, and then just as the virus would have used our machinery to convert the DNA to a protein, our cell would use that DNA, make the protein, and then this protein will somehow be shown on the surface, and therefore our immune cells will recognize it. So this is a strategy and is extremely good in theory. The only drawback is that to date, none of the vaccines that are actually in use have been able to use this strategy. Another strategy that people have used and which actually may be very useful this time is what they do is they take the shell of another virus, something like our flu virus or measles virus, things that we have been exposed to earlier. But again, they alter it enough so that they will not cause a disease. And then they put the DNA of the spike protein into this virus and then inject the virus, right? So it's like it's sort of a disguise. You're sending your protein, or at least the code for the protein, in another organism which our body is used to looking at and taking it out. And then it is hoped, and that strategy is likely to work again extensively. So this has been used for the Ebola vaccine, and it did work. But unfortunately, by the time they had this vaccine in place, the pandemic, it wasn't really a pandemic, the disease had reduced enough so that we didn't have to immunize a large number of people. So that is the other strategy. It's going to be a challenge. There is no doubt about it. But as I said, because everybody has been affected by this all over the world, the idea is that if a vaccine does get developed anywhere in the world, it's likely to be used by all the countries. And I'm sure that all the heads of states and the countries would definitely work towards. So in fact, we are actually, so if you've heard of an institution called the Serum Institute of India, which is located in Pune, Pune is actually or rather it has already got the go ahead to actually make a particular vaccine, which is being developed by Oxford University in UK. I think if I can backtrack just a little bit, let me tell you how a vaccine will get developed. Right? So these are strategies. As I said, this is what works in the lab. So first thing is you have to check that whatever strategy you're going to use will work, in the sense that the cells will be able to generate an immune response. Before using anything in humans, you have to first make sure that it is safe. Very, very important. Usually when a vaccine is developed, the safety is measured first in animals. So you start with things like mice, then go to monkeys, and then finally you would actually, assuming that it has worked in these animals, you would try it for humans. And even in humans, there are something which we call as phased trials. The first trial is done with maybe 50 or 100 people just to make sure that the vaccine is safe. And that is usually a healthy person who volunteers for the trial. If it is safe, then there is the next round of trial, where they would actually do it in a population which is likely to be susceptible. and then see whether it really has an effect. Normally, this phase 2 trial would last for a year or two to be able to really be convinced that it is working. Here, of course, I'm sure they will short circuit it and go into production. So if this works, then normally they would go into a phase 3 trial, which is where you throw it open to maybe 10,000 people or more and see the efficacy. Because making a vaccine is an expensive process. If it is not going to work, then why would any institute or company put in probably billions of dollars or rupees in it to make a vaccine, right? But this time I think all these rules have been put. So this group in Oxford has tried it in monkeys and we've seen that it works. They are reasonably confident it will work. So I think they've done the safety trials as well. So they are reasonably confident in working humans. So they actually told certain companies like the Serum Institute of India to start making bulk vaccines. So if it is effective, we may be actually in a good situation to get vaccine, because our country is one of the places where the vaccine is actually going to be made. And as I said, I'm sure the government is going to pour in money in this particular situation to ensure that people, well, they probably start with the higher risk people and then at some point try and immunize as many people as possible. It may never be possible to immunize every Indian. So you remember there was a Spanish flu, what they called in about a century ago. We didn't have a vaccine at that time, but people survived. So unfortunately at that time, because we had no treatment and no vaccine, I believe something like a third of the population died. This is what we want to avoid this time. So what do we mean by this herd immunity? Right? So we have to understand. As I said, what happens is that a person gets the organism. It's not necessary that the person is going to always come out of the disease. In this case, most people do, even if it's a mild form. Okay, the organism, the immune system in many people will be able to fight the disease. Right? What does that mean? That means this person has now become immune to the disease. Right? Now, if this person gets the virus again, they will definitely not get the disease. They've got the immunity. The idea of this herd immunity is that if there are, say, 100 people in a particular place, okay, and the virus is spreading, but out of that, over a period of time, say 80% or 80 people have got immunity, then what happens? The virus can't spread because it has very few people who can infect anyone further. And remember, a virus will not survive unless it infects another person. So that is what we mean by herd immunity. That if sufficient people are able to build their own immunity through exposure to the organism. So some are naturally immune, some will get immunized with a vaccine. Okay? So once that happens, then what happens is the virus doesn't really get a chance to, or at least it doesn't have enough of the population to infect and spread the disease. And that will happen. The fear is that currently, because we don't even have a treatment, we don't have an effective treatment. So what is happening is that everybody who falls sick, or at least gets exposed, and actually starts showing the symptoms, and if they have other complications, their life is under threat, which means they need hospitalization and they need medical care, which many of us are not going to be able to cope with. And that is why most of our governments have used this idea of social distancing, and because we don't understand this concept, they've imposed this lockdown. Lockdown ensures that we will interact with very few people. And those people, therefore, who do get infected will either get immune or will get the medical facilities and will be able to recover and come out. As I said, what happens is that once a person either recovers, will definitely would have an immune response. Okay, one of the ways the immune response plays a role is it makes an antibody, which is a soluble component, it's a protein, which can then recognize the virus next time and will trigger elimination of that particular virus. So what people are using is that what they're doing is they're taking the serum or plasma of patients who have recovered, which means they have the antibody, and they're using it as a therapy, not for vaccine. So that means if a person already has the virus, till the time that that person can make their own antibodies, you are providing them or supplying them with artificial antibodies. So it is being used as a form of therapy. And it has worked many times. But as you can imagine, look at the number of people who have recovered. How many of them would be able to donate plasma? How much of that will you be able to give? So it is a very limited form of treatment. Ideally, if we had a very effective drug, we might have been in a better shape. But unfortunately, we don't really have a very effective drug against it. And again, though hundreds of labs are trying out various things, it's not a sure shot method of recovery therapy, right?
Thank you, Dr. Yasmin. I like how you always leave us with a hopeful message, even in these dark times. It was really a joy to speak to you.